So this is a part 4 video of GATE 2023 exam revision reloader series. Okay. So first we'll be studying general properties of histone. So I think you all know about histone. Histone is a type of protein that is present in your chromosome and they are positively charged basic protein. Okay. So they generally consist of more number of lysine amino acid and arginine amino acid. Since this histone is a positive charge and our DNA is negative charge, this histone play an important role in compact packaging of chromosome. Okay, so this histone is a basic structural functional unit of your nucleosome. Nucleosome play a vital role in the structural support of chromosome. The histones are classified into core histone and linker histone. Core histone include H2A, H2B, H3, H4, which will be forming a octamer in case of nucleosome. So each of this particular compound, each of this histone will be replicated twice. So there will be 2 H2A, 2 H2B, 2 H3, 2 H4. They are forming a compact octamer of nucleosome. Linker histone is named as H1. The molecular weight of H2A is 14,000. Molecular weight of H2B is 13,900. The molecular weight of H3 is 15,400. The molecular weight of H4 is 11,400. The molecular weight of linker histone is 20,800. Next, you should know the percentage of lysine and arginine amino acid present in them. Regarding to H2A, the total number of percentage of lysine and arginine amino acid is 20%. In H2B, it is little bit higher, 22%. In H3, it is 23, and H4, it is 24. And next in H1, it is present highest amount, that is 32% of lysine and arginine amino acid residues are present in linker histone. Next, we are going to study three major class of transposable element. First is DNA only transposon. Okay, the structure of DNA only transposon is that it consists of only short inverted repeat at each end, and next one is retroviral like retrotransposon. So retrotransposons are classified in two types. One is retroviral like and another one is non-retroviral retrotransposon. The structure of retroviral like retrotransposition is that they will be having a directly repeat, a long terminal repeat, that is LTR repeat at each end, whereas the structure of non-retroviral retrotransposition is that they will be having a poly A tail at 3' end of RNA transcript, and most commonly the 5' end of this particular retrotransposition is truncated. Okay, and next the enzyme required for movement of this particular gene is, with regard in DNA only transposition or DNA transposition, the enzyme required is transposase gene, whereas regarding to retroviral like retrotransposition require reverse transcriptase enzyme and integrase enzyme, and next category, that is non-retroviral retrotransposition, they require reverse transcription and transcriptase enzyme and endonuclease enzyme. In retroviral retrotransposition, they require integrase enzyme, and in non-retroviral retrotransposition, they require endonuclease. Okay. The mode of movement of this DNA only transposition is that they move as DNA either by cut and paste mechanism or replicative pathway, that is copy and paste mechanism. Whereas with regarding to movement of this retroviral like retrotransposon, they move via an RNA intermediate produced by a promoter in this LTR. Okay, this long terminal repeat consists of promoter, so they will be producing a RNA intermediate. So they require a RNA intermediate. Whereas with regarding to non-retroviral retrotransposition, they also move with an RNA intermediate, but most commonly this RNA intermediate is produced from a neighboring promoter. Okay, and the example of DNA only transposition include P element of Drosophila, ACDC element of maize, and TN3 and TN10 element of E. coli, and TAM3 of Snapdragon. Next, the example of retroviral like retrotransposition include copy element from Drosophila, TY1 of yeast, THE1 of human, and BS1 from maize. Next is non-retroviral like retrotransposition. The example include F element from Drosophila, L1 element from human, and CIN4 element from maize. So this is again I had given you few important things. So here we are going to study only the example. The example for bacterial replicative transposition include TN3, gamma 8, phage move, and the example of bacterial cut and paste retrotransposition include TN5, TN10, TN7, IS 911, TN 917. And the example of eukaryotic transposition include P11 from Drosophila, marinary element, hat family element, and TC1. And next is RNA mediated transposition. So first thing is with the example for virus like retrotransposition include TY element of yeast and copy element of Drosophila. Next example of poly A retrotransposition include F and G element of Drosophila, lion and sign element of mammal, and ALU sequence element of human. Okay, so you can study either this one or else you can study this one. Both are equivalent only. So this poly A retrotransposition is an example of this non-retroviral retrotransposition, and this virus-like retrotransposition is example of retroviral-like retrotransposition. Next is we are going to study secondary structure of protein. So there are three secondary structures present in protein. One is alpha helix and beta sheet and collagen triple helix. The example of alpha helix include alpha keratin of your hair, feathers of birds and nails. Next example of beta conformation include silk fibrion. Next is collagen triple helix include collagen of tendons, bone and bone matrix. Next, we are going to study few inorganic ions which are serving as a cofactor for various enzymes. Okay, so what is mean by a cofactor? If you ask me what is mean by cofactor, so the cofactor or a non-proteinous compound which will be helping for the enzyme in doing catalytic reaction. Okay, so we'll be seeing the ions, inorganic ions that are serving as a cofactor for what enzyme, like that we are going to see. So first is Ce2 plus ion which is a cofactor for cytochrome oxidase. Next is Fe2 plus and ferric ions, Fe3 plus ions. These are acting as a cofactor for cytochrome oxidase, catalase and peroxidase. Next is potassium ions, K plus ions which are used as a cofactor for pyruvate kinase enzyme. Next is Mg2 plus which is used as a cofactor for hexokinase, glucose 6-phosphatase enzyme and pyruvate kinase. Next is Mn2 plus which is acting as a cofactor for arginase enzyme and ribonucleate reductase enzyme. Next is MO molybdenum which is acting as a cofactor for dinitrogenase. Next is nickel which is acting as a cofactor for urease, and next is selenium which is acting as a cofactor for glutathione peroxidase. Last one is zinc which is acting as a cofactor for carbonic anhydrase, alcohol dehydrogenase and carboxypeptidase A and B. Okay, so these are the list of inorganic ions that are acting as a cofactor for different enzymes. Okay. So now we will be seeing many enzymes. They provide a critical coenzyme for human cells. So here we are going to study list of vitamins that are acting as a coenzyme. Okay. So first we will be studying. So coenzyme is again an organic molecule which will be binding to the active site of certain enzyme, and they will be also helping in the enzyme catalytic reaction. These are the list of vitamin, and this is the active form of vitamin, and these are the derivative of vitamin. So vitamin themselves they can't able to act as a coenzyme. So they need to convert either into an active form or a derivative. Only then they can able to participate in different enzyme catalytic reaction. Okay. First is this thiamine, vitamin B1. The active form of this particular vitamin is thiamine pyrophosphate. And next is riboflavin, that is vitamin B2. The active form of this vitamin or a derivative of this vitamin is FADH. And next is niacin. The active form of niacin is NADH and NADPH. Next is pantothenic acid. The active form of this enzyme is coenzyme A. So coenzyme A is active form of the vitamin pantothenic acid or vitamin B5. Next is pyridoxal phosphate, which is a coenzyme of pyridoxine, that is vitamin B6. And lipoamide is a coenzyme form of lipolic acid, which is again a vitamin. Okay. Next is tetrahydrofolate, which is a derivative from folic acid. Next is cobalamin coenzyme, which is a derivative from vitamin B12. Okay. So next thing we are going to see the list of enzyme catalytic reaction requiring these coenzymes. So first is thiamine pyrophosphate, which is involved in activation and transfer of your aldehyde group. Next, both this FADH, NADH and NADPH all are involved in oxidation and reduction reaction. Next, coenzyme A is involved in acyl group activation and transfer of acyl group. Next is pyridoxal phosphate, which is involved in activation of amino acid, and also this pyridoxal phosphate is regulating an important enzyme, that is glycogen phosphorylase, which is involved in process of glycogenolysis. Next is biotine acting as a coenzyme for CO2 activation as well as transfer of this carbon dioxide group. Next is lipoamide, which is again used in cell group activation and also oxidation reduction reaction. Next is THF, which is involved in activation and transfer of single carbon group. Like this tetrahydrofolate is acting as a coenzyme for those enzymes that are involved in synthesis of nucleic acid, synthesis of amino acid, synthesis of polyamide. Okay. Next is last one is cobalamin coenzyme, which is acting as a coenzyme for those enzymes that are involved in the process of isomerization and methyl group transfer. Okay. So the next video will be continuing. Thank you friends, thanks for watching this video.